all, what is excretory system? This is a system which is responsible for excretion of waste product from body. What are the waste products? All the extra metabolites, uric acid, extra electrolytes, they all are called waste products, as well as water. The extra water is also excreted from body as a waste. Enzymes, small proteins, etc. Right? What are the processes which are involved in this excretion? First of all, juxtamedullary or ultrafiltration, glomerular filtration. The second is tubular reabsorption. The third is tubular secretions. Right? But behind these three processes, a lot of processes are working. A lot of hormones are responsible. A lot of electrolytes are responsible. Right? What are the exact percentages which are reabsorbing in proximal convoluted tubules, or what are the characteristics of the cells which are present in proximal convoluted tubule and distal convoluted tubules, as well as in collecting ducts, because the nephron have different types of cells. They have a lot of different types of cells. For example, in Bowman's capsule and glomerulus, they have fenestrated epithelium. In Bowman's capsule, they have podocytes. In proximal convoluted tubules, they have brush border cuboidal epithelium. In loop of Henle, they have simple cuboidal epithelium. In distal convoluted tubules, nephron have principal cells. In collecting ducts, they have principal cell as well as intercalated A cells and intercalated B cells. And the transportation and mechanisms occur in all over this nephrons is different from each other. Right? Because, and one thing another, that distal convoluted tubules have very special types of cells which are called macula densa, and they are responsible for detecting the sodium concentration in distal convoluted tubules. Right? We will focus on three things as well as with this nephron. First is isotonic solution, second is hypotonic solution, third is hypertonic solution. In some areas of the nephron have hypertonic solution, and some areas they have isotonic solution, and some areas they have hypotonic solution. And this hypotonic, hypertonic and isotonic solutions are responsible for this secretion and reabsorption. Secretion and reabsorption. First of all, what is the difference? What is the actual difference between the reabsorption and secretion? For example, I have, this is the tube from nephron, and this is blood capillary. Right? For example, from here, when this green molecular substance is going to blood, this process is called reabsorption. And when these things are coming from blood into nephron, this is called secretion. What is filtration? When the things are passing from a mesh-like structure. What's mean by mesh? Mesh galbel tawai. Chan ya galbel ni awle chiji chanai. This is called mesh. When the things are filtering from a mesh-like structure, this is called filtration. Why it is called ultrafiltration? Because all the macromolecules are filtering from glomerulus. So they are called ultrafiltration because all the things are filtered from the glomerulus into the Bowman's capsule. Right? And some of the things will 100% reabsorb from proximal convoluted tubule. Right? So first of all, we will focus on a brief view on nephron. What is nephron? Nephron is a structural and functional unit of kidney. They have two parts. Nephron is divided into two parts. One is renal corpuscle and renal tubules. Good. Renal corpuscles is divided into two parts. One is glomerular, second is Bowman's capsule. Right? This cup-like structure is called Bowman's capsule, and this is the proximal convoluted tubule. But right, if at least some amount of or some length of straight tubule, right? It will called the straight proximal tubule, and these are convoluted, so they will called proximal convoluted tubules, right? The next part is loop of Henle. Loop of Henle have two parts. Mainly it have two parts. One is descending limb, one is ascending limb, and it forming distal convoluted tubule, and these distal convoluted tubules are ending by collecting ducts, right? So for example, here I have, this is renal artery, and from renal artery it is divides and divides and divides and forms a glomerular capillary tuft. This is called glomerular capillary tuft, and this is efferent arteriole, right? Here are, for example, for this is the efferent arteriole and this is the efferent arteriole, right? For example, this blood is coming from renal artery, right? So the parts in this part of the glomerulus, we have a very special types of cells here. They are called mesangial cells. They are called mesangial cells, and a very special type of membrane is present here, and this membrane is called glomerular basement membrane. This is very important because it has heparin sulfate, which makes us very negative, and the negative things cannot filter from here because they are refilling each other. This is so simple, right? This efferent arteriole is further divided into peritubular capillary. So for example, this is the division of and this blood is running with the tubules of nephron, right? This is the simple structure of nephron. First of all, this is the efferent arteriole. This tuft-like structure is called glomerulus, and these blue cells are called mesangial cells. These mesangial cells are very important because it squeezes the glomerulus for full filtration. It also forming some connective tissues for support, right? And this arteriole is called efferent arteriole, and further this efferent arteriole is divided into this green structure. For example, this is simple structure, and these capillaries are called peritubular capillaries, right? This blue structure here, this is called proximal convoluted tubules. This structure is called Bowman's capsule, and Bowman's capsule is funnel-like, having funnel-like structure, and this funnel-like structure is very important in capsular hydrostatic pressures, right? This area is called the straight proximal tubules. This is just the part after the Bowman's capsule. This U-like structure, this structure here, this is called loop of Henle. But for this is anatomical structure. For physiology, we will remember that this limb is called descending limb of loop of Henle, and this part, this green part, this red part, this is called the thin part of ascending limb of loop of Henle, and this part is called thick part of ascending limb of loop of Henle. Why we are separating these areas? For mechanism, because the mechanisms occurs here and the mechanisms occur here are very different from each other. Also the mechanisms occur here are different from proximal convoluted tubules, and the mechanisms or the physiological functions occur in Bowman's capsules are different from proximal convoluted tubule. Also the osmolalities. What is the osmolality of blood? It is about 300 millismol per liter. Right? Remember it with yourself, because we are going to discuss this in urine formation. 300 millismol per liter. This is the normal osmolality of blood. We will compare the osmolalities between the blood and the nephron. Right? And this part is called distal convoluted tubule generally, but for physiological functions we are dividing this distal convoluted tubules because the mechanisms of physiological functions occur in distal convoluted tubules are different from each other, in early as well as in late. So this part is called early distal convoluted tubule, and this part is called late distal convoluted tubule, and this part is called collecting ducts. Now, anyone who can tell me which type of cells are present in different parts of the nephron? Is there a need of discussion for the cells? The cells are the kam cells pa kam ki. Yes, sir. So, for example, I am taking this nephron again, right? This is not only important for your paper, it is important for your whole medical life, because when you are giving some drugs or medicines to a patient, you will take care of the kidney failure, the kidney diseases, the nephron dysfunctions, etc., GFR, NFE, right? Because more drugs excrete from body through urine, and when you are not taking care of the drugs excretion, you will harm the patient, right? So this topic, this renal system is very important for your whole medical life. This is not necessary that all the things will come in paper, right? So for example, here I have, this is the afferent arteriole, and this afferent arteriole is further divided into different types of different capillaries. So I am taking one capillary from this structure, right? The first cell types in glomerulus. This is glomerulus, right? This is glomerulus. Glomerulus capillaries have a very special type of epithelium, and this epithelium is special because they have a lot of penetrations. They have a lot of holes. Not all the capillary cells or epithelium in body have holes in their cytoplasms. This is only for making this area mesh-like, right? And the diameter, you know all, the diameter of this hole is 70 to 100 nanometer, right? So first of all, glomerulus have, for example, this is glomerulus. First of all, it have penetrated epithelium. Penetrated epithelium. What is penetrated epithelium? The epithelium having 1,000 and hundreds of pores, right, for filtration. This is called penetrated epithelium, right? The second thing, a very special types of cells are present in this glomerulus called mesangial cells. And these mesangial cells, collectively, it is called mesangium. It is called mesangium, and it forming a special type of connective tissues which maintain this area, also squeeze or forcing the glomerulus for filtration because they are contractile, right? So these are the cells. First, fenestrated epithelium. Second, mesangial cells, right? The next thing is the basement membrane. So for example, this is the basement membrane. Which type of basement membrane? Glomerular basement membrane, because alveoli also have basement membranes and they are called alveolar basement membrane. This is called GBM, they are called ABM, right? So here I have told you they have triple alpha helical chains which are formed of collagen type 4. This is formed of collagen type 4, and at the end they have non-collagenous part of glomerular basement membrane, right? So for example, I am taking this area from this glomerular basement membrane here. For example, this is the glomerular basement membrane and they have a dark 3 alpha helical structures. Right, this area, this is called lamina rara interna. This area is called lamina rara externa, and this area is called lamina densa. Why they are important? For filtration. And if we neutralize the charges on the basement membrane, it will cause the hereditary glomerulonephritis, right? And these are the non-collagenous parts of the basement membrane. So this is basement membrane. These cells are negatively charged. This membrane is also negatively charged. The next, we have Bowman's capsule, and Bowman's capsule have, changing this marker, these are called podocytes. These cells are called podocytes, and this layer is called visceral layer of Bowman's capsule, and this layer is called parietal layer of Bowman's capsule. Make this very small. These are called parietal layer, right? So the next cell types are of Bowman's capsule. Cells in Bowman's capsule. First one is podocytes. These cells are called podocytes, and these cells are called, these are simple epithelium. Simple epithelium, and this layer is called the visceral layer, and this layer is called parietal layer. So in Bowman's capsule, podocytes are present in visceral layer, and simple epithelium are present in parietal layer, right? Next, we have proximal convoluted tubule. So for example, I am not making it convoluted, but this is, for example, proximal convoluted tubules, and this proximal convoluted tubules have a very special types of cells because they have brush borders on luminal side, and on basolateral side they have plain basement membrane, right? So next thing is proximal convoluted tubules, and this proximal convoluted tubules have which type of cells? Brush bordered cuboidal epithelium. What is the purpose of this brush border epithelium? It increases the surface area for reabsorption, but it have a very special characteristic, these cells, because they are very highly rich in mitochondria, and proximal convoluted tubule cells They have a lot of mitochondria, because all the transporter present here which are important for absorption and reabsorption will use energy, and the energy will come from mitochondria, right? This is the next part is loop of Henle. For example, this is the loop of Henle, and all over the loop of Henle we have simple cuboidal epithelium. Simple cuboidal epithelium, but there is a lot of difference between the ascending and descending limb, right? We will discuss that these proteins and these cells have a very special types of transporter which are called aquaporine type 1 transporter, and this is only permeable to water, and this area is only permeable to electrolytes. Which type of electrolytes? Sodium, potassium, chloride, calcium, magnesium and potassium, right? Because the calcium will be repelling by potassium. This is a very unique type of transport here which occur due to repelling of two charges, right? So this is the loop of Henle, and loop of Henle have which type of epithelium? Simple cuboidal epithelium, right? The next part is distal convoluted tubule. The distal convoluted tubules have cells, cuboidal like cells, but they are called principal cells, distal convoluted tubules, and this distal convoluted tubules have principal cells. They also have some of the cuboidal epithelium and some intercalated cells, but basically they have principal cells. And in last we have collecting duct, and in collecting duct we have like this type of arrangement. This is principal cell. These all red cells are principal cells, right? For example, this is principal cell, and these green cells are called intercalated cells. If you got an MCQ in your paper that intercalated A and B cells are present in, generally it is present in kidney, and kidney it is present in nephron, and nephron it is present in collecting ducts, right? Why it is called intercalated? Because they are calated between two principal cells. It is sandwiched by two principal cells. So these red cells are called principal cells, and these principal cells are working like this principal cell. But these green cells are called intercalated cells, type 1 A cells, and these are called intercalated B cells, right? And in last it have simple epithelium which is highly permeable to urea, right? We will discuss this. So these are the cell types present in nephron, right? Any confusion? Now we will go for the mechanisms occur in all the parts of nephron, right? The cell types it has to be